everyone, my name is Naphat and today I'm going to talk about the research in the topic of MMS19 links cytoplasmic ion sulfur cluster assembly to DNA metabolism. So due to I have just only 15 minutes talk, so in the talk today I will focus on only the techniques that they use to find the answer about the function and how MMS19 links to cytoplasmic ion sulfur cluster and how MMS19 assembly or take part in DNA metabolism. So when we talk about DNA metabolism, it includes DNA replication or the process that reproduces information on DNA strand. And in this in this process, it requires like almost 100% accuracy. And DNA metabolism also includes DNA repair and recombination, which is the process that preserves the current information and fix the information when the damage occurs. And it also includes the DNA degradation or the breakdown or degrade of DNA strand. And in each process here requires DNA metabolism proteins, which is the function of DNA metabolism proteins depends on the ability to coordinate or interact with ion sulfur cluster proteins. So for ion sulfur cluster, we have talked about ion sulfur clusters many times. And ion sulfur cluster is the ubiquitous cofactor that composed of ion and inorganic sulfur. It takes part in many processes like electron transport chain, regulatory sensing, photosynthesis, or DNA repair. So for ion sulfur cluster, biosynthesis can take part in both mitochondria and cytoplasm. And the deficiency in biosynthesis of ion sulfur cluster can lead to many diseases. But in this paper, in this study, they will not focus on ion sulfur biosynthesis, but they focus on another proteins that take part in DNA metabolism, and that protein is MMS19. MMS19 is the heat repeat protein that involved in DNA metabolism in DNA repair process, especially for nucleotide excision repair or NER, and it also takes part in RNA polymerase II transcription and coordinate with other protein, which is TF2H, RAD3, and XPD, which I will talk about TF2H and XPD more in the next part of my talk. And the function of MMS19 that they found from previous study, they found that MMS19 functions as the interaction partner, like a main interaction partner of RTEL1 or the regulator of telomere length. So RTEL1 regulates the telomere length and also maintain genomic instability. That means we know that genomic instability can lead to many diseases like cancer or neuromuscular disease. So the objective of this study is to investigate how MMS19 protein links to cytosolic ion sulfur proteins and how can it involve in DNA metabolism. So the importance of this study is we can further study and use the result from this study to develop the new approach for targeting diseases, for treating diseases that have been caused by genomic instability. So the main techniques that they use in this study is co-immunoprecipitation or co-IP. Co-IP, the main idea of immunoprecipitation is it is like a pull-down experiment that we try to immunoprecipitate the intact protein complex by using specific antibody and that specific antibody that against specific protein target to form the immune complex and pull down all the complex together. So you will see from this picture that when we have the protein mixture here, we will find the specific antibody that's specific to our protein of interest or MMS19, and we will capture the antibody and protein of interest, and if the protein of interest or MMS19 interact or cooperate with other proteins or interaction partner, they will all together form the complex here. And we will capture that on the beads or resin, the purple circle here. And then we just elute other proteins in the protein mixture. We just wash other protein in a protein mixture, and we will get only the complex between beads, antibody, MMS19, and the protein that acts as interaction partner. So in this step, we just elute only the protein of interest, MMS19, and the interaction partner. And then we just analyze the results here by using just the basic molecular method like immunoblot or Western blot to analyze the protein-protein interaction.
So Pierce, this company Pierce always makes this look very easy, right? So when does this technique have problems? When is it difficult to use this technique?
The disadvantages of co-IP technique is the non-specific binding between protein and protein. It's difficult to detect, and also we use the antibody here. We can get like antibody contamination, and also the ability to like maintain the interaction between protein like MMS19 and other proteins is hard to maintain that all during the process of co-IP, because we use the elution buffer. We use many chemical and mechanical states to this process, so maybe we lost some protein interaction, and we can get like contamination from antibody.
I think that's the biggest thing. When the affinity between, when the protein-protein interaction affinity is weak, this technique runs into problems.
Yeah.
Or if it's a transient interaction, this technique runs into problems.
The transient interaction.
There are ways around that, just for people that want to do that. You can cross-link your proteins and then IP them.
Yes.
So that's a way to make a transient or weak complex stronger.
Stronger, and we can detect from co-IP.
Also, if the interacting partners, if they're almost of similar molecular weight, how do
you detect
Yeah, exactly.
Yeah, no, that's true. Yeah, absolutely.
Well, is this supposed to be like a one-stop shop? I mean, or is this just another purification?
It's not just a purification. It's just like pull down, and it's like if you talk about only just immunoprecipitation, you will just find the protein that's specific binding to the antibody. But for cold immunoprecipitation, you can use this technique to detect the protein that interact to your protein of interest.
It's more to ID than anything else. So, for example, in Leanne's talk, where it's unknown factor that may bind, co-IP would be one way to.
Yeah, I was thinking about that.
If it's a protein and it's got decent affinity and everything else.
Okay. And they also use the mass spectrometry techniques as a quantitative techniques to to find out how much interaction partner cooperate with the MMS19, and they also use the liquid scintillation counting and use the radioactive ion 55 to label ion in ion sulfur cluster and check how MMS19 involved in ion sulfur cluster biosynthesis. That I will talk more about this technique in the data analysis part. And here's my suggestion. They can also use other techniques to check the interaction between the interaction partners. Like, for example, yeast-2 hybrid techniques. For yeast-2 hybrid techniques, the main idea of this technique is we have two plasmids. One acts as base here that contains DNA binding domain, and another one acts as prey here that contains activation domain. So when base catch prey, they will form the complex here that contains DNA binding domain, activation domain, and RNA polymerase all together, and that active and turn on the transcription of reporter genes. So if we transform the base and prey into yeast cells, and we use the reporter gene that can encode essential amino acid like histidine or leucine, something like that, that's very important for yeast growth. So we can detect that from the growth of yeast cells in the medium that lack of essential amino acid. Here's another suggestion for this study, to study the interaction between interaction partner of MMS19. So here's come the data analysis. The first part of data analysis, they study the interaction between MMS19 and RTEL1 protein. So for RTEL1 protein, we already discussed about that. RTEL1 involved in the genomic stability. So they first study with MS, mass spec, and they found that MMS19 is the most abundant interaction partner of MMS19 of RTEL1 protein. And then they use reciprocal co-IP to confirm the result from MS. So they use the whole cell extract from HEC293 cells. As you can see from the picture A, the reciprocal co-IP is the antibody that they use as the x-axis over there is the antibody that they use to pull down the MMS19 and RTEL1 from the fraction, and the antibody here, anti-MYC and anti-MMS19 is the antibody that they use in Western blot to detect specific here, and you will get a bands on the Western blot here. So from this result, from the reciprocal co-IP, it can confirm the result from mass spec that, okay, MMS19 interact with RTEL1 protein. And so now they know that MMS19 indeed interact with RTEL1. So where are these proteins in the cells? They overexpress the RTEL flag in cytoplasm, nucleoplasm, and chromatin in HeLa cells. And then they do the reciprocal co-IP again, and they found that the MMS19 and RTEL1 are mostly found in cytoplasm fraction. So they can conclude that MMS19 and RTEL1 is are cytoplasmic proteins.
How they did that? I mean, how they overexpress, say, that protein in cytoplasm in the chromosome, and what are they NP? How did they do the experiment?
I'm not sure whether they explain that or not.
In a specific location, I mean.
Expression के लाग सीधे करते हैं क्लोनिंग को
I'm not sure whether they discuss about that in the supplemental data or not, because I just focus on the main paper. I didn't run through the supplemental data, but I think they explain more about the procedure that they use or the method that they use more in the supplemental data, but I didn't go through that.
I've never done cell work because for you guys, you did